Thank you, Anderson. Can you all hear me? Well, perfect. Okay, you may need the headphone if you don't, but yeah. So today in this session, we are going to discuss about something called a golden batch, or identifying the best recipe when you are producing chemical compounds. So in pharma or life sciences, when we think about different product that we manufacture, right? It could be chemical compounds or medical devices or prosthetics or biologics such as regenerative medicine or cell medicine, right? Uh, each of these product type could leverage SAS to optimize the overall manufacturing process to improve some target or KPIs, right? So it could be your quality of your product, the quantity of the product, total time it takes to manufacture something, or the total cost of your overall production. The reason I list this in this particular use case, right, will showcase how SAS via platform can be used for designing penicillin manufacturing procedure, but the same analytical concept is applicable to all these different types of products, right? So you can think beyond just chemical compounds. So we are in a very tightly regulated industry, right? So we have to follow the GMP process, right? Good manufacturing process. And that process needs to be validated throughout the life sciences manufacturing cycle, right? And there are three aspects of this. One is process design, right, where you are kind of defining the parameters, how you do the manufacturing. You are maybe doing many different experiments and setting those manufacturing parameters. Then how the process, like the production, how it's performing, right? So we call it process performance qualification. So you evaluate the design, right, and do different test control strategy and test the reproducibility. And then once you have set all these process parameters, okay, you are good to go for production. And at that point, you may want to do continuous process verification, right? So you are verifying if your process is correct and within the standard deviation of the parameters that you have defined. So at SAS, we can support all these three aspects. For this particular use case, we are going to focus on number one, okay? So the process design part, okay? So this will be our focus, but before I jump into that, I really want to zoom out a little because it's not just about manufacturing. Everything is connected, right? So your manufacturing is connected to your supply chain. You may have multiple plants, right? And at SAS, we can always think about an integrated optimization. So again, today we are focusing on a single plant, a process design process, but we can always connect this to multiple plants or supply chain. Now, in a normal manufacturing setting, you may have a plant, and within that plant, you may have some sort of bioreactors, right, where you do your large scale production. And these bioreactors have some process parameters or controls, right? And many of these controls are already automated through PIDs or other mechanism. But often we see there are some manual control parameters that are set by an operator, right? So we see that operators are kind of fine tuning these parameters to find out what is the best parameters or controlling them, okay? So once you do that, then you go into your production. So this is when you are in your final manufacturing stage. But even before we go there, right, there is a process design phase, right? This is where your R&D team must decide on what are the optimal ingredients, what are the optimal environment or process for your manufacturing steps, right? We often call that the pilot stage of production. So this is before you scale up, you do some experiment with small batches, right? And in this type of pilot production, you may want to control your oxygen level, pH level, temperature, different ingredients or catalysts, right? And you may also want to optimize for time, like either faster production or when you are getting your maximum yield. So almost all manufacturers, right, follow this process, and what we see among our customer, they often have historical data of batches, but they are not organized, right? So they are not managed properly, and oftentimes they are underutilized and not always databased, okay? So what we are going to showcase, how we can take historical batch information, right, of your pilot production stage and do some exploratory analysis first, right? So we want to identify what worked well or what went wrong. From there, we are also looking to do some prescriptive analytics, right? So can we train some models that would be able to prescribe us what a future scenario might look like, right? And can we recommend optimum control for your KPI? So this is how the demo background is set, right? So you can think of a product R&D or process engineering team has collected this historical data from batches of production, okay? And there are many control parameters that affect the quality and quantity of the batch. And we are going to do some exploratory analysis and identify key factors from there. And then we are going to train some model for predictive analytics. So the data that we have used in this particular scenario comes from a paper written by Goldrick. They made available 100 batches of data that we use to train machine learning models. Okay? And this is very similar to what you find in real manufacturing plant. Okay? Like a bioreactor, which have many, you see, inputs going in, and you can, through those knobs, you can control the flow. In this case, we have 7 manual controls that were driven by operators, okay? And there are many automated controls that are already determined by PID. And they can measure the penicillin concentration every 12 hours, right? So that's our output measurement against which we can optimize the process, okay? So some of the business question you can ask when you have this type of data, right? Which recipe historically produced the highest concentration of penicillin, right? Because we want to harvest the penicillin at highest concentration to get maximum yield. You may ask what were the drivers of the higher yield or quantity, right? Can we identify them quickly in a data-driven way? Can we predict outcome of new recipe? That means, can we simulate a scenario that was not present in the historical data, completely new, and are we able to, through using the model, predict what the yield might look like if I were to run such experiment? And then can we prescribe certain recipe for achieving a target result, right? So it could be, in this case, we are focusing on yield, but you can also focus on your total cost, right? So your KPI can change time to time. So the beauty of SAS Viya platform is, once you have the data, right, all the downstream process we do within SAS Viya and platform. Okay, so starting from your data management, ETL, visualization, then building the models, and using that model to simulate and optimize the scenarios. Okay, so again, we start with the exploratory analysis, and I'm going to show you how a typical dashboard might look like, right? So this is all the 100 batches, right? So we collected the data, and we are now doing exploratory analysis through our dashboard, right? And this is an interactive dashboard, so you can select a single batch, or you can select multiple batch altogether, right? And you are seeing different trends, right? And you see the peak of each curve occurs at different time points, right? And this is dependent on the input scenario. We can drill down to specific batches, right? So now I'm drilling down into one particular batch, and I want to highlight a time point where the maximum concentration has occurred, because that is important. To optimize the yield, we want to reach the highest penicillin concentration in the shortest amount of time, right? That's why you are optimizing your cost and getting the highest yield. On the bottom, you are seeing some input parameters as well altogether. Now from this graph, we can take two values, right? One is the maximum penicillin concentration, that's one value, and another is the time point at which hour this maximum concentration is happening. We can take these two values and plot them into a scatter plot, right? So in this scatter plot, along the x-axis, you have time required to reach the maximum concentration, and the y-axis, you have the maximum concentration itself, right? So if we just think about yield, right? So the rightmost, that would be your best batch, because it's giving you the maximum penicillin concentration. But unfortunately, it's taking you about 280 hours, right? So if you look at the rightmost, sorry about that. If you look at the rightmost, right, this is the batch that has given me the highest concentration, but it took 280 hours. So that might not be the optimum, right? On the other hand, you'll see there are some batches that are potential candidate of worst batch, right? So like this one here, you see, it ran for almost 240 hours, but it did not give enough concentration, right? So you may want to know as a domain expert what went wrong for that particular batch, right? So from here directly you can drill down and you do further diagnostics, okay? So again, this is where as a domain expert, you are making a decision based on your historical data, okay? So far we have not done any modeling, it's just exploratory analysis, but it's already giving you some insights, and you are trying to understand based on my historical data, what were the best parameter settings that we could use for future production. Next stage, we start some modeling, right? So we use SAS Vias auto explanation. So this is basically bunch of random forest model looking at the target, the response variable and the underlying factors and giving variable importance ranking, right? So these are the factors that are impacting our yield the most, right? And you see, like for example, the top one is the aeration rate change. So it's in a completely data-driven way, we are identifying which parameters are influencing our yield the most. Again, if instead of yield, if you are looking at a different KPI, you'll get the variable ranking for that particular target response, okay? So this provides some insight. So what often we see that domain experts have empirical knowledge, right? So they already know which parameters might impact the yield. But at the same time, through the data-driven process, they identify some additional parameters that they never thought of, right? So this is where, one, we validated that we are seeing what is empirically already known. We are seeing that. At the same time, we are identifying additional parameters that might impact the yield. So from here, now we want to build a predictive model, okay? So what we do, we take our historical batches of data, split them into training and testing set, so holdout set, right? And we use SAS Model Studio to train machine learning models. And in this case, we trained multiple models, including generalized linear model, decision tree, and random forest. And we can do model comparison, right? And you can do it either in a programmatic way or in a low-code, no-code way, based on your choice. Once we have identified a champion model, right, we validate it against a test or holdout batches, right? And here you can see we are comparing how the model is performing against an holdout batch, right? Whether it is completely rubbish or because this now I am testing the model against a data it was not trained. That's why it's a holdout set, right? And it gave us enough confidence that we found that for most of the holdout set, the prediction was within the 95% confidence interval. So we are very happy with the model training. So once we have trained a data-driven model, now this model allows us to do in silico simulation, right? Because now I can take this model and partner with different combinations of input, right? That was not present in the actual data. So now we are simulating completely new scenarios, right? And oftentimes we'll call it digital twin. Because we have a model that represents the bioreactor now in a data-driven way, right? Now we can perturb all these different input parameters and see the predicted outcomes, right? So in this mini video, you're seeing a domain expert now can kind of create their own synthetic pilot run, right, by tweaking all these different parameters instead of running an actual experiments, right? So quickly, now they're able to find insights. So what would happen if I were to change the oil flow pattern, right? What is the deviation I can expect to see? And based on that, you can actually design your actual experiment. Now, since we have it as a model, right, we can simulate all these different scenarios in an automated way, right? And now we can look at the distribution of the concentration over time across all these different model runs, right? And from there, we can select or identify the best synthetic batches, right? So just to remind you, we started with the historical batches, and here you are looking at completely synthetic batches, right? Now, oftentimes, what the model predicts may not be physically or practically relevant, right? But that's where, as a domain expert, you would decide, because model might recommend some input parameters that not physically viable, right? It may destroy the whole process. But that's when you can go back to the dashboard and see what are the input patterns the model is suggesting, and you can make decision as a domain expert. So this is for identifying highest yield in a given batch. Now what we see within an organization, you may have different KPIs, right? So these are all the results from the synthetic batches. So this particular recipe ID 4486 is giving me the highest batch yield, right? So this is for one given batch. But you might be interested in highest annual yield, right? So the reason you may ask, okay, why the highest batch is not giving me highest annual yield? The reason is quite simple, because if you look at the time, right? So the batch that gives me the highest yield runs 192 hours, right? And in this particular scenario, every time you run a batch, there is a 24-hour downtime for the bioreactor, okay? And then at the end of year, there is a 30-day downtime just to maintaining the bioreactors, right? So there is a 30-day downtime at the end of year, and after every batch, you have 24-hour downtime. So we incorporate all these factors. Based on that, we calculate the annual yield. Now, you may say, okay, I don't care about the maximum. As long as I'm getting 100,000 kilograms in a given year, in the shortest amount of time, I'm happy, right? In that case, you may choose this particular recipe, right? So again, this is just to showcase your target might be highest batch yield, your KPI might be slightly different, right? You may have an overall annual goal. We can address all these things within the platform. So at a high level, this is what I wanted to present. If you have any question, we have some time for question, and I am here at the HLS booth. You can come if you want to see the demo live. I can walk you through. Thank you.